Good evening, everyone, and welcome to Manchester Cafe Sci Online. Judging by the numbers tonight, we seem to have found a successful adaptation, as it may be some time before we can all sensibly gather in person. We've even been able to insert some extra events into the programme. I have the option to extend the Zoom hosting for a year at a discount. If you'd like to support that, you can find the virtual sweetie jar at PayPal.me/Win Cafe Sci, which I will copy into the chat for the meeting. It also enables us to stream live on YouTube for people who aren't keen on Zoom, and we can post recordings on our YouTube channel as well, where you'll find a recording of Janus's talk on space debris from last time, and all the links will be on our website at wingcafesci.org.uk. After tonight's talk, we'll take a break for about 10 minutes before the Q&A. Please put any questions you have for Andrew into the chat, and I will read them out when we resume. Our virtual venue tonight may be familiar to some of you as the Jenner House Museum, home to one of the great pioneers in immunology. Tonight's speaker will be familiar to many of you and has previously given some very clear and helpful primers, basic concepts in biological sciences, particularly some of the more recent developments. Tonight, he's going to outline a topic of great current interest. What is immunity? So please welcome AndrewSeber.
Thank you, William. Thanks for the opportunity to have a chat with you about this. My background is that of a teacher rather than a current research scientist. My interest is getting basic concepts over, because there's so much in the news about this area. That it's sometimes you can get the advanced stuff, but the grounding just isn't so easily available. So I'm going to describe to you what immunity is, how it's used, and what it means for us. My opening slide is really illustrating the point that we are assailed by pathogens and other invading substances and organisms all the time. At the moment, we've all got viruses, we've all got bacteria, and our bodies are dealing with it as best they can, usually with great success. Occasionally things don't go quite so well. So I'm going to move on to my second slide. By the way, if you sort of have a particular slide that you want to comment on, I'll let you know the number. This is slide number 1. Slide number 2, a little bit of the etymology for those people who like this sort of thing. The term immune actually came from people who are in Rome who'd been soldiers, had done their military service. Once they'd done it once, they weren't asked to do it again. But in the 19th century, the term became used for the sort of way in which we use it now, that you've had a disease and you're not going to get it again. I've already mentioned Edward Jenner, so I'm going to say a little bit about him because that's always very interesting. He came to fame because of what he did in promoting the concept and practice of vaccination. This is the sort of famous background story to it, and the red, which you probably can't read too well, says, "Where are you going to, my pretty maid? I'm going a-milking, sir," she said. The origin of that is that milkmaids tended to have really quite good complexions. In the 18th century, about 10% of the population had smallpox, and the death rate was enormous. Disfigurement was quite common. So if you had the pox, and well, there are various forms of pox, but this particular kind, smallpox, was deadly, widespread, and absolutely devastating. But for some reason, milkmaids seem to get that rather less. And the famous man who we all hear about is Edward Jenner. And this is a famous picture showing him doing something to his gardener's son. Ethics were slightly different in those days, and it wasn't quite so complicated to get through ethics committees and so on before you started experimenting. The thing to bear in mind about Edward Jenner is that he had himself been protected against smallpox as a young man or probably as a child. But the way that it was done was to give people actually the smallpox virus, the process called variolation. And Edward experienced this, and it was pretty risky. because although it would potentially protect you against getting smallpox later on, it also carried the severe risk of actually getting the disease. So he was very keen to try and find some way of avoiding that. And he looked at Sarah Nelms, who was a local milkmaid. Noticed she had a bit of a pock on her hand, but nothing serious. And she said she thought she'd caught it from a cow. So what Edward Jenner did was he took some pus from that mark on Sarah's hand and injected it or scraped it into young Mr. Phipps's arm. And then a little while later, he came back and he administered smallpox toxin to him. He actually gave him the smallpox virus. But bear in mind, he might have done that anyway because he would have been trying to protect him through variolation. That was quite common. To his great relief, he found that the boy did not get smallpox. Having been given a taste of cowpox, he didn't contract smallpox. So Edward then wrote off to the Royal Society. They said, more evidence. So he did it to about 23 other people. That had already been done. There were some farmers who already tried this and found it seemed to work. So then it began to catch on. The actual name, vacina or vaccina, vaca means cow, for those of you who know Latin, I don't, so I'm just guessing. But the reason why that name is used is because it was the cowpox as opposed to variola, which is the general name given to smallpox. So, brilliant idea. So brilliant that it caught on internationally. This is a famous sculpture. You can't see it locally, unfortunately. You have to go to Rome. You can't go to Rome, can you? No one can at the moment yet. And it's showing Edward Jenner inoculating his own son, age of 11 months. He gave him the cowpox because by then he'd decided that not only was it safe, it was also a hell of a lot safer than taking the risk of getting smallpox disease, sorry. And so it caught on. And in fact, the government took it up. And in 1840, it outlawed variolation and said, if you want to be protected against smallpox, you need to be vaccinated. And so people all over the world started to get vaccinated. Early in the 19th century, Napoleon had all of his troops vaccinated, and he gave Edward Jenner a medal. We happened to be at war with France at the time, but never mind. It seemed to be worthwhile. So all over the country, people started to be vaccinated. It wasn't universally popular. There were some people who thought this was dangerous. So now, the boy on the left, his parents were influenced by a campaign mounted by his local MP in Leicester at the beginning of the 20th century. This is photograph taken in 1900 in Leicester. The boy on the right, his parents didn't go along with their local MP's view that vaccination was a terrible danger, and you can see the outcome. It's a pretty horrific picture. I think it illustrates just why there was so much effort put in to dealing with smallpox and why vaccination became such a brilliant idea. The idea was extended quite a lot to other diseases. And this slide shows a picture. This is now slide 8, by the way, if you're counting. This shows the graphic representation of how various vaccines, as they now became universally called, were effective. I say universally called because actually some of these diseases, well, none of them actually use cowpox. What they're doing, they're using some form of the virus which causes the disease in some changed form, killed, modified, only parts of the virus used. You can see the top thing shows diphtheria, where a vaccine was introduced just before late 1940s, late 1930s, and how the disease went down. You might say, well, it looks as if diphtheria is going down anyway, but that, of course, was just the lull where there would have been almost certainly another spike later on. Polio, if you're of a certain age, and I have seen the pictures of some of you, and like me, you probably went off and had polio with a needle, not on a nice sugar cube back in the 1950s. and that had a huge effect on reducing polio, which I know some of my contemporaries actually suffered. Measles vaccine came in the 1960s, and again, the incidence went down significantly. Opposition to vaccination didn't ever go away, and this is just to illustrate a number of points. One of the purposes of vaccination is clearly to protect the individual, but it also protects the whole of our society. This is where we get this term herd immunity. If you look on the left of this graph, which is back in 1997, well over 90% of children were receiving the MMR vaccine. By about 2004, this had gone down. And this happened periodically, where there were people saying that there were dangers involved with the MMR vaccine, so other parents got concerned and decided their children wouldn't be vaccinated. And those are the bars showing how many. So by about 2004, only 80% of people were taking up the MMR vaccine. And you can see the line shows what happened. But once people had stopped taking it up, the incidence of these illnesses, measles, mumps, and rubella, went up. And that is largely because once you got below about 90%, the herd immunity was no longer enough. The way it works is that if people are resistant to a disease because they are immune to it, they're not going to catch it and pass it on. So if nearly everybody is either resistant because of having had the illness or has been immunized, then there's not enough people to pass it on. Obviously, it has big resonance for today for other reasons. So that's a bit of background to vaccination. I want to talk now, and this is really the purpose of my talk, to give you some idea as to how the body is doing this. I think it's fairly well understood that you have an illness, you therefore have resistance to it, therefore you're not going to get it again. But the question I wanted to delve into is, what is this resistance? How does it actually work? The picture I'm showing now, which is slide number 10, I'm simply showing a diagram. On the left-hand side, in the yellow box, I've got the word external. We actually resist the invasion of our bodies by obvious things, skin, and that's a hugely important barrier. Very, very little can actually get through your skin. There are weaker points. The mucous membranes, which line your respiratory system, which line your alimentary canal, mucous membrane, it has to be wet so that either food or gases can be exchanged. Your lungs are lined with mucous membrane. That membrane has the substance mucus is capable of resisting the entry of quite a few potential pathogens, but not all. So where there are risks, it's where you've got that sort of place, eyes, nose, mouth, lungs, gut, urinogenital system, where things can get in. Your nasal hair, again, as you get older, you get more of it, don't you? Good thing, because it does help to keep some things out. So that's your first kind of barrier, which is a sort of passive barrier. Then, moving to the right-hand side of the diagram, the internal system. And this is where the cells of the body actually come into operation. And these cells are carried in the blood and in other parts of the body. And again, I'm showing there are two lines, and I'll go over this in much more detail. On the left-hand side, in probably blue or purple, first line, there's a group of cells which will be the first responders to invasion. Second line is a more sophisticated response to that invasion, which is something we're able to do because we are higher animals. This next slide, which is number 10, is number 11, sorry, slide 11. This is a microscope slide, magnification about 500 times or so, not difficult to do. In my teaching days, we were allowed to get students to prick themselves with needles and put drops of blood onto microscope slides and look at it. but my own blood looked pretty much like this. And there are two main types of cell. I know this will be something that many of you know very well, just worth just going over it and just take me a few seconds. The two main types of cell are red blood cells, which clearly are those that look pinkish in this diagram. And they, you notice, have a sort of a light bit in the middle. That's because those cells, uniquely almost amongst our cells, have no nucleus. The other cells are coloured blue. They are actually clear and transparent in nature, but in order to show them up, the slide is infused with a dye, a blue dye, and the white blood cells take up that blue dye and appear blue. So in diagrams, you always see white blood cells are coloured blue. They do have nuclei, and they come in various shapes and sizes, and they are our defence against pathogenic organisms. Now moving on to slide 12, which I'll use more than once during this talk. All of these cells are made in the bone marrow. So your bones, we always think of as being structural substances which are there to form a skeleton, to give a shape and size. But actually, in the inside is not wasted. It's hollow, but within it you have bone marrow. And you will have heard of bone marrow transplants, because that's where we have stem cells. Again, stem cells, term used a lot. These particular kinds of cells are called hematopoietic cells. They're blood stem cells. Moving down the diagram, on the left, I'm showing a myeloid progenitor cell, and on the right, a lymphoid progenitor cell. So the stem cell differentiates during development as you grow in the womb in order to be able to produce these two broad types of blood cell. The myeloid on the left will give rise to red blood cells, some of them, to platelets. Platelets are fragments of cells which are used as plugs when there is a physical injury. They will go to the site of a cut and will help to plug the gap along with various other substances. Then in the box under my myeloid progenitor cell, I've got a whole bunch of cells. There's a neutrophil, an eosinophil, a basophil, and a monocyte. Don't worry about the details of those names. These are all types of white blood cell. They have slightly different functions, but they are all concerned with the so-called innate immune system. They will respond quickly to any invader in a fairly broad brush way. On the right-hand side, we've got the lymphoid progenitor cell, which gives rise to lymphocytes. And I'll come back to that later. So now what I'm going to do, I'm going to move on to my next slide and say a bit more about that left-hand box, the innate immune system. Innate or natural immunity. Nothing unnatural about any of this. What this is about is detecting anything that's foreign. The blood cells, the white blood cells, can tell whether when they encounter something, it's from you or it's from an invader. Doesn't matter the details of what it's like. If it's foreign, it's going to try and do something about it. And what these cells will do is identify the foreignness, destroy them, and get rid of them. There's a process called phagocytosis, and that means cell eating. So very crudely, it is a crude process, when there is something which is foreign and shouldn't be there, these white blood cells will go and eat it. There is a consequence of the action of white blood cells, which is known as inflammation. It means just what it says. It's inflammation as you and I know it. I'll come back to the cellular details of that. Those white blood cells can also communicate with each other and with other cells, and can also prepare for other cells to destroy the foreign invaders. There's a substance called complement, which is used to destroy anything that's invading. So I'm now moving on to a picture, which is slide number 14. And this is a picture taken under an electron microscope. Very high magnification, much more than a light microscope. And that's been coloured in, just to make it more obvious. The rod-shaped orange bits on the left are anthrax bacilli. So if those get into your body, then they will produce serious poisons and bring about death. The yellowy, shiny, luminous thing there is actually a monocyte. It's one of the white blood cells. And what it's doing, it knows, it can tell that that is not part of your body, and it's going to eat it. It's a smallish cell. It's used to be called a macrophage, and it will come along and it will gobble up those foreign substances. So as you get invaded, the first line of defense is to try and eat anything that's coming in. When you've got a very small, low level of invasion, that might be enough. And at the moment, you are being invaded. You don't know it, but you're breathing stuff in, you're touching surfaces, and you've probably been eating something. And you are being invaded. And this front line of defense will deal with quite a lot like that invasion. The inflammation that I mentioned comes about because not only do these cells go and eat the invaders, they send out chemical signals to other parts of the body in order to enhance the effort of getting rid of these things. So you will see if there has been something's got into your body. For example, I recently had a toe that had been injured and it got red and hot and puffy. And what was happening there was that the chemicals produced by the white blood cells were making the blood vessels, the capillaries, expand. So they were no longer as liquid proof as they normally are, and blood would therefore flow into the infected area. In fact, blood plasma, the white colourless part of the blood, would flow out into the infected area. So that's where the swelling comes about as well. So it gets hot, it gets swollen, and you get pain because there are substances released again by the blood cells. Pain is a good thing, so that the fitness people tell us. But pain in this case is a way of informing you that something is wrong and you should do something about it, like try and stop people treading on that swollen toe. Okay, so here we have a schematic diagram. This is my 16th slide, and it's showing there's a spike going down into it. That's something that's come into the skin. It's probably introduced bacteria, same as red discs, and the white blood cells are secreting a whole cocktail of chemicals called cytokines. And I'm mentioning this name now because cytokine has a particular importance in the sorts of diseases that we're worrying about now and struggling with now. Cytokines are the chemical messengers that will expand the blood vessels, will enable white blood cells to get through, and will start to raise temperature. Now, in fact, one of the consequences of the raised temperature
Sorry,
is
baby, I'm my mum.
Somebody online there. Okay, so one of the cytokines will actually cause your hypothalamus in your brain to allow your body temperature to increase. The reason for that is that if your body temperature increases, it will make you, as a host, less hospitable to invading bacteria. There is a risk, because if your temperature goes up too much, not only will you be less hospitable to the invading bacteria, you will be less hospitable to your own cells, and they will cease to function. But of course, the risk of doing nothing is death. Doing something is possible discomfort, but hopefully response to and repulsion of these invading substances. So just to sort of recap that, the cytokines are the things released by the blood cells, the white blood cells, and they have a whole range of effects, including allowing body temperature to rise. The cytokines also make other cells increase, so it'll stimulate other white blood cells to come along and start acting and to produce other substances. Normally, once you've dealt with the invasion, that subsides. However, sometimes it doesn't, and this is called a cytokine storm. One of the things that has been discussed in the current work on COVID, that some people react in this way. It's also thought that people with particularly active immune systems, normally a good thing, might be at risk of a cytokine storm. That emerged from the 1918 Great Flu epidemic, where a lot of people who were most seriously affected were young, fit, healthy people, often coming back from the army and so on. Whether that's the true reason or not, I don't know, but certainly a cytokine storm is really bad news and means that action has to be taken. One of the white blood cells that is particularly important is known as the macrophage. I mentioned the small blood cells being macrophages. They do a bit of eating. These are the big eaters. These are the heroes of the immune system. So I'm very keen on the macrophages. And what they do is to eat big time, but they don't actually completely digest what they have eaten. And they preserve parts of the invading pathogen and present them on their surface. So now they're going to be able to stimulate activity elsewhere. And that's where I'm going to move over to the other kind of immunity. The first kind I talked about was natural or innate immunity. This is adaptive or acquired immunity, much more specific. This will not only decide whether something is foreign or not, it'll decide exactly what it is and how it's going to get rid of it. One of the aspects of acquired immunity is the production of antibodies. And these are proteins that float around in the plasma. They are humoral, humor meaning fluid. And so they're going to be in the plasma and will be able to help to fight against anything that comes in. There are also some cells delightfully known as killer cells. And as the name suggests, they will go and kill cells that are potentially dangerous. Most of the cells they kill are your cells, but there are safeguards. Okay, so I'm now moving from the innate or natural immunity to adaptive or acquired immunity. The cells that are most concerned with this are called lymphocytes. Slightly more enlarged versions is slide 20 now of my blood slide, and showing one of the cells enlarged, a large nucleus, and it's called a lymphocyte because it lives in part of the body known as a lymph system. It's one of these lovely diagrams that you never know what on earth it's meant to be showing. But as well as your blood system, which most of us are quite familiar with, which has arteries and veins carrying blood around the body, there is a lymph system, and it consists of a number of tubes and drains which house most of the white blood cells that you see occasionally in blood, but in great quantity in lymph. There are things called lymph nodes, and those are areas where the lymph vessels form a sort of a knot and can act as a filter. During times of infection, those lymph nodes can get swollen. So you may find that you've got an infection which is diagnosed in part by feeling the lymph nodes. There are the inguinal lymph nodes at the top of the leg, and they can become swollen, particularly when there's some infection in the leg. Ones that people are most familiar with are right at the top, the cervical lymph nodes in the neck, and that's what gets swollen when you've got mumps. And what's going on is that the white blood cells have congregated there and are mounting a big attack on whatever's invading you. Lymph joins up with the blood, it drains into a vein in your neck. So these two things are not separate, nothing is separate here, everything is integrated. So going back to my original diagram, here I am, I'm over on the right-hand side now, and what I'm looking at is the bit which has the adaptive, it's the box in the bottom right-hand corner, the adaptive system. You can see that there are a few crossover lines. This is because the systems do communicate with each other, and cells are useful in both contexts. Now, when I looked at that box, I'll go back to it, lymphocytes, T lymphocytes and B lymphocytes. The B lymphocytes make antibodies. The T lymphocytes are more concerned with infected cells. So I'm going to start off by talking you through what the B lymphocytes do. Their job is to make antibodies. Antibodies are fantastically important. The antibody has a particular property, and that is that it has a shape which will make it lock on to something which is foreign. And in this case, we use the term antigen. Now you'll hear this term used quite a lot now, antigen testing. What you're trying to find out is what is the thing that is actually going to cause the problem. The antibodies will stick to the antigen, and one of the useful things it will do is they will link up with each other. So if you've got something invading you, bacterium, virus, or whatever, and there are antibodies in your blood to that invader, it will make them clump together. So no longer are they able to get into your cells, they've been joined up, and a nice macrophage, bottom right-hand corner, number 3 in the picture, will come along and eat it. So you've got a virus invasion to which there are already antibodies, then the macrophage will eat them and they will disappear. So again, eating very important. Now, this is a picture of an antibody. Again, fantastically high magnification. In fact, it's more than that. It's partly enhanced by a bit of artistry. And this is a curious Y-shaped molecule. All the blobs you can see there, the tiny blobs are atoms. And so we've got thousands and thousands of atoms making up this protein molecule. It's also called an immunoglobulin. Immuno, because it's concerned with the immune system, and globulin, because when they first discovered them, they thought they looked rather globular. So you'll hear this term immunoglobulin used a great deal in discussions in immunity. I'm going to simplify this picture because you can't really make out what's going on in the detail, apart from pointing out the 2 arrows are pointing to a shape at the end of the top parts of the Y. The 2 arrows are showing a sort of little dent. Now that dent or cleft or whatever is the business part of the antibody, because that is going to be complementary, just as a lock is to a key, to an antigen. Simplified diagram now. There are 2 protein chains that make up an antibody, and they're duplicated. So you've got left-hand and right-hand side of this molecule are exactly the same. On the top, you've got some antigens. These would be protein molecules or something like that, which would be produced by a potential invader. And they're all of different shapes because each invader will have its own particular characteristic. But this antibody will only lock on to one of those antigens. They are fantastically specific. They will be able to join up with and stick to, and therefore neutralize that particular shape. So this antibody will only get onto that blue shape. It won't touch the red one or a pair of pants or whatever else is in that diagram. The antibodies will stick to the antigen, but they will focus on a particular region. They're so specialized that they will focus on one particular part of the antigen. The term used for that is epitope. Again, you don't hear that quite so often, but in some of the scientific journals, they will talk about an epitope, which is the shape to which the antibody will bind on the antigen. So here's a picture of a potential antigen with various bits on it. Just happens to be COVID-19. There are a few diagrams of COVID-19 around at the moment, which is why I've latched on to this one. And it's actually a surprisingly simple structure. It's got a skin, which you can see here, with bits sticking out of it. The S protein stands for spike, and that is part of the COVID virus molecule, which is going to stick onto the cell and enables it to get in. So what we would like to have, we would like to have antibodies to at least the spike and preferably other parts of that virus molecule. This virus molecule is absolutely minuscule. You cannot see it. It's below the resolution of a light microscope. You might see a dot, but this is, again, derived from an electron microscope. If you look at it in 2 dimensions, it looks like a crown with spikes coming out of it, hence the name coronavirus, because it's crown-shaped. Now, the cell that I mentioned, the lymphocyte, which is going to be really important here, is the B cell. So the B lymphocyte has on its outer surface, on its membrane, some antibodies sticking out. This B lymphocyte that I'm showing here can only make that kind of antibody, can't make any other, and it sticks out of its surface. And this antibody will join up with one particular antigen. And here we have, it's probably a virus molecule, it's got some spiky bits sticking out of it, and this antibody will stick and grab onto that particular shape, which is great, because now you're in business, because that antibody will be made in quantities. Now, what I'm showing you here on the left hand of the picture is a whole load of different B cells. And your body is making these B cells all the time. It's like a little factory, knocking them out, producing them over and over again. You've got something like 2 trillion B cells in your body. So fantastically high number. But only one type of those 2 trillion will be the one that's going to bind onto this particular potentially invading molecule. So showing on the left-hand side some antigens, which are little dots. They don't stick to number 1, 2, or 3, but will stick to number 4. Once that antigen is bound to that B cell is going to form a clone. In other words, that number 4 cell is going to reproduce itself over and over and over again in high numbers very quickly and produce some things which are called memory B cells and some which are called plasma cells. The memory cell is crucially important. It's going to remember the shape. It's going to remember that that antibody it was making was really useful, and it will retire to the lymph system and wait. And as you can imagine, it'll wait until the antigen comes along again. Some of those clone will form so-called plasma cells, bottom right-hand corner. And the plasma cell doesn't just make the antibodies, it releases them. It lets them flow out of its cell body into the plasma of the blood and all around the body. So what happens is that once the B cell is able to detect an antigen, it will make antibodies to it, and they will get into your body and provide an immediate defense. You can actually use this therapeutically. You can harvest these antibodies and you can inject them into an infected person. And those antibodies will actually help to stop the antigen or the pathogen getting into the body and causing harm. The only problem with it is that that's short-lived. It'll only last as long as those antibodies last. What you really want is the body to make its own antibodies. And that's what vaccination is all about. It's persuading the body to make antibodies, set up a memory so it can deal with them in the future, release some antibodies straight away so it can deal with any immediate problems. Question is, and someone just flashing up a quick question then, how on earth can you have so many antibodies? And this mysterious diagram I'm putting up there just to mystify you for a while I work out how I'm going to explain this. What I'm saying is that any antigen that comes into your body has never seen it before. Your body will be able to make an antibody to that antigen, normally. Okay, someone's going to give me an exception, but that's the normal process. So you've got to be able to make a huge number. You've got to be able to make millions and millions and millions of different antibodies. Those of you who came to my talks on DNA will know that we've got genes, bits of DNA. Each bit of DNA will make a specific protein. We've got 20,000 genes. So how on earth, with just 20,000 genes, are you able to produce millions of unique antibodies? This diagram, again, don't worry about the details. I'm just showing you bits of DNA. On the right-hand side of the diagram is something called the constant region. That's the part of the antibody, which is the fat bit at the bottom, which is the same generally for antibodies. So that's going to be the basics. The next three regions to the left vary. And what happens is that when the white blood cells, the lymphocytes, reproduce, they break the rule. Normally, the rule is when a cell reproduces, it makes a completely faithful copy of the original DNA. But in the white blood cells, the lymphocytes, that is allowed to change. And what happens is there's part of that gene which is allowed to go bonkers. It will produce loads and loads of different variations. And that way, you're able to produce a huge number of different types of cell, each capable of making one antibody. Sounds pretty amazing, but there it is. The immunoglobulins that are produced have various descriptions, and I'm only showing this because you'll hear different descriptions. When those B cells are first maturing, that they're growing in your lymph system, and they pick up the antigen, the first type of immunoglobulin they produce is the one in the bottom left-hand corner, the IgM, which is immunoglobulin M. Nothing more magic than that. And those are pretty good. They are able to form clumps and bind things together. The things they're not produced in such quantity as the one at the top left, and that's the really important one, the IgG. This is the gamma globulin. And you'll hear reference to the gamma globulins. These are the ones that are produced in huge numbers and flow out into the plasma and will be your first line of defense. It's useful because when you do an antibody test, and some of you may have had this done for COVID-19, if there's a lot of IgM, it means that the infection is recent because the blood cells are producing the M type. If it's IgG, then that means that the infection is a bit older and you've got on to the big production of the gamma globulins. There I'm showing that the IgM are particularly good at agglutinating. So, okay, I'm coming towards the sort of the final stage of what I'm going to explain. I'm going back again to my original diagram, and I'm still on the adaptive box on the right. And what I've talked about so far have been the B lymphocytes, which will give rise to plasma cells, which will give rise to antibodies. You've got another set of lymphocytes called the T lymphocytes, and they have a number of functions. Broadly speaking, your B cells, the B lymphocytes, will attack invaders outside the cells. The T lymphocytes will attack the invaders once they've got inside the cells. Particularly important, this one, for viral infection. This is one of my favorites again. This is a lovely macrophage on the left. And this is taken under a high resolution light microscope. And what it's doing, that the cell on the left is a cytotoxic T cell. It's eating the cell on the right. And the reason it's eating it is because it is found that that cell has got some foreign substances. The cell on the right was originally a human cell, but it's been invaded by a virus, probably in this case. Therefore, on its surface, there are indicators that it's no longer only your cell. So the nice friendly T cell will come along and eat it and destroy it. The way in which viruses get into the body is many and varied, and this is why so much research is going on at the moment into the absolute detailed structure of the COVID-19. What this is showing at the top of the diagram, these are our pictures of COVID viruses, which consist of RNA, that's the genetic substance in the middle, surrounded by a bag of mainly fatty sort of substances on which are these proteins stuck. The spike protein happens to be the same shape as something which can bind to an ACE receptor. Some of you are on ACE inhibitors. I probably will be by the end of this talk. And what ACE is an angiotensin converting enzyme, big name. But what it is, it's a protein that is normally on your blood cells, particularly those lining the blood vessels and the lungs and kidneys and various other parts of the body. In this case, lungs most important, helping to control your various functions, including blood pressure. But the virus happens to have this spike, which is the right shape to bind onto that receptor. So it sticks onto it, and the cell thinks this is a friend. And it brings it into the body and says, come in, you're welcome. The virus then releases its RNA, which takes over your protein-making system. And instead of being dedicated to making your proteins, it makes virus proteins. And then they will be released from the cell and off they will go. And this is just showing the same thing in just a little more detail. So the invader enters. And the reason why some viruses are particularly problematic is because they have this ability to enter the cell in large numbers and wreak havoc. So going back to the overall shape of what I've been saying, two systems, innate immunity, rapid response, but won't tackle the details, adaptive immunity, slow response, very specific, and will get at the thing that you really want to get at, the disease-causing organism. I mean, there are lots of military analogies on this because, and it is really like that what you've got is you've got an invasion, and so you send your frontline troops out to do what they can, but you don't know what the enemy is like. You know it's an enemy, that's all. You know that it's not part of you, so you will try and destroy it. But you don't know the details of the enemy. So you've sent out battleships, submarines and so on, but this organism actually is an aircraft carrier. So you're going to need to know specifically what sort of airplanes has it got. And so your adaptive system spots the specificity and can respond to it. And the B cells are going to produce antibodies. The T cells also have those antibodies on their surface, but they are going to be used to do the killing. On that right-hand side, I've got two lots of T cells, CD4 plus and CD8 plus. Again, don't worry about the details. Is that the 4 plus is the one that is going to actually help other cells. It's called a helper cell. It's a bit of sensible terminology there. And what that does is it signals to the B cells to produce more antibodies and various other things. It switches them on and can also switch them off. And the switching off is very important because you don't want the thing to go awry. The other one, the 8+ cell, that is the cytotoxic cell. That's the one that's going to do some killing. There are also memory T cells. So what I've painted is quite a complex picture. Just to make you feel better, it's not half as complex as the full picture. But I really want to get hold of is the idea, you've got these two systems, it's the B cells that are there producing antibodies, the T cells help them, and the T cells will tackle cells that have been infected. Viruses generally will get into cells, and the T cells will come and kill your cells, but it's worth it. You want to kill the ones that are on the surface so they don't get any further. The reason why we get ill is because the system can get overwhelmed. A mild dose of an infective agent is usually dealt with without even knowing it. If it's a big dose, then it can overwhelm your system. And in serious cases, it overwhelms it totally, and the system just really can't quite cope, and therefore you get ill. I'm going to go back now to where I started, which is our friend the cowpox. On the left-hand side, I'm showing a picture of a cowpox virus. There it's got its DNA, it's got its genetic material inside it, and it's got spikes on the outside, just like any good virus would. Smallpox looks pretty similar. The smallpox and the cowpox virus, under electron microscope, you would think they're really very, very similar indeed. However, when you look in detail, and you can look at this picture, the nobbles, the proteins are on the outside, are not quite the same. But some of them are. So what happens is with vaccination, The cowpox induces antibodies against cowpox surface antigens, some of which are the same as those on the smallpox. So that's why vaccination works with an organism that causes one disease, cowpox, can tackle another one, smallpox. That idea is also currently important in research into how you deal with COVID, using what you know about the SARS virus, which was around a while ago, didn't kill quite so many people. So that's really the outline of what I wanted to say. I know it's a bit of a gallop, but just wanted to make sure you got that overall picture. What I'm going to do now is pause. You're going to cook up questions. If they're very difficult, I'll refer them to William, but I will do my best then to answer any of those questions. If you want me to go back over something, feel free. I can make the presentation available to you. I think William's got it in PDF form, so it can be sent by anyone who wants it. And I'm always happy to answer questions. So I could send you my email address if you want to ask me anything. Okay, I'll leave it there. Thanks very much for listening. I hope you're still awake, and we'll come back shortly. Thank you.
Thank you, Andrew. And where are we? So let's get some good questions into the chat after the break. Before we break, a quick heads up on our next talks. On 6th of July, we have Professor Patrick James from the University of Southampton, and his talk is Rezoning and Livable Cities. The talk will look at what makes a place and how we can engineer cities to deliver on our aspirations. We'll discuss city disruptors and how they are accelerating change in our cities, COVID-19 being foremost amongst them. Shrinking retail, walkability, the value of green space, etc. And on the 20th of July, we've rescheduled from April Alan Ratner's talk on languages of the world, identifying the language of text. We'll start the Q&A. Oh, so Steve asked, how close are we to creating a vaccine against COVID-19?
Very close. There are already vaccines being produced. However, producing a vaccine and knowing that it is going to work and be safe to use is another thing. Normally, you'd reckon on 18 months to 2 years to produce a vaccine, and they're galloping along trying to get it done more quickly, using some of what they've learned from SARS, using the huge amount of knowledge they've got of the structure of the vaccine. But however much you know, you can't go ahead unless you test, because a novel virus, by its very name, means that it's not one that we worked with before, and therefore you really can't be sure what's going to happen. So they're hoping to get something this autumn, but a lot of fingers are being crossed, and there are concerns in case we go up ahead too quickly.
Yeah, I've registered with the National Institute of Health Research Biobank with the University of Southampton, and they've invited me to participate in the Oxford vaccine trial. I haven't heard any more since I said yes. I think perhaps 3 likely outcomes. One is they'll decide that I'm not in contact with enough people to be a useful participant. 2 is that they'll do a blood test and find that I've already got antibodies, as I'm pretty sure that I've had a mild form of it. That will be interesting to know in itself. Or failing that, I'll either be on the control group or the test group. The control group, they're administering a meningitis vaccine, and the test group are having the COVID vaccine. So watch this space on that one.
Yes. Well, the early stages, they want young, healthy volunteers. So obviously you qualify in both counts, William, so you're all right.
Well, I certainly did. Well, it's qualified as being healthy enough anyway. I think it's people under 65 they were looking for, so just about count as far as that's concerned. Might even, I don't think there was a lower age limit.
Right, what
So-
other questions are coming in?
Okay, so from Tony and Jenny, does a few virus bodies just get killed by the rapid response, so you do not get any antibodies? Does a few more help? Then there's too much that overloads the secondary response?
I mean, the general principle is that a large infection is more likely to cause an overload. The first line of defense will tackle viruses to some extent, but viruses don't have on their surface the sort of indicators that bacteria and other pathogens have. So it means that you get fairly quickly into the second line. So cells will get infected, and you need to be able to tackle them at that stage. If you think what's going to happen is a cell's going to get infected, it'll produce surface antigens, macrophage will recognize them, gobble them up, and go and report to other white blood cells, including B cells that produce the antibodies. Once the B cell is activated and it's producing the antibodies will be in the body fluids, particularly the blood, and will therefore make the virus particles clump together as soon as they're spotted. So what you really want is to have primed B cells which are producing antibodies in the body fluids that will stick to and neutralize the virus particles. The killer cells will kill the cells that are infected, but you really want to get at them before that stage.
Tony and Jenny, does that answer your question though? Because it was there were sort of several parts to it. Did we cover that?
Yes, I was trying to really get at, I suppose, can you give someone a very small amount of COVID-19 without them getting the full disease?
Yeah, I mean, what the aim of a vaccine is to produce a version of the pathogen which is not itself going to cause disease. So it can be attenuated, which really means just slightly modified. The Salk vaccine, which was used to treat polio, was basically a killed version of the vaccine. Or you can chop the virus up and take parts of it, and those spike proteins for a particular target. But a lot of it, you've really got to do experiments to work out what's going to work. It's often the case that if you use part of the pathogen, the response requires boosting later on. If those of you like me who had the smallpox vaccine, you had to go and have a booster every now and then and carry around with you a certificate to say that you'd had it to be allowed into quite a number of other countries. So yeah, the inoculation is a small dose of something which has been made safe, but will still generate antibodies.
Andrew, have you seen much research on the concept of viral load that I've seen talked about? Because in the context of COVID-19, it was being suggested that some healthcare workers were getting it much more severely because they were in close contact with infected and infectious people, whereas other people who perhaps just had a brief social exchange might have got off more lightly. I don't know how solid that view is.
The whole issue around COVID is that it is a novel virus. Although we've got a lot of examples to work with, we haven't had a lot of time to see how things develop over time. I mean, the general principle is the bigger the dose, the more likely you are to be infected. There are also very many other factors. I've talked about the innate and the adaptive system. They are not the same from one person to another. We are all genetically different, and therefore we have different extents to which our responses will be more or less efficient. So you can't generalize. There's nothing in here which is automatic. So some people are going to be more vulnerable than others. A lot of the work into ethnicity is that looking at whether there are clusters of people with particular characteristics make them more vulnerable. But this stage, a lot of it is still, to put it sort of generously, hypothesizing, and put it more literally, it's guesswork. At one time, they thought that the ACE2 inhibitor was going to be a way of determining whether someone was more or less vulnerable, but it looks more complicated than that. The problem is that the virus seems like a simple thing, but the reaction we have to it is hugely complex.
Robin asked what percentage of viruses have been successfully combated with vaccines. I suppose that has the rider of the viruses that are pathogenic to humans.
Yes, the pathogenic one, because most viruses just are around and don't do very much. Most viruses are not specific to humans, but there are diseases for which no vaccine has been produced. I mean, this is one of the things to bear in mind. We can talk glibly about a vaccine, but you can't guarantee that there will be a successful vaccine. I believe that there will be. Looking at particularly the Oxford research, it's very encouraging, but there are absolutely no guarantees in this business.
Henry's asked, am I right in thinking the reason why plasma from a previously infected person only works in the short term in someone else is that the antibody cells are not recreating themselves?
Correct. That's exactly right. If you had my son had to go off to India at very short notice, and so there'd be no time to vaccinate him against certain illnesses. So he was given a dose of gamma globulins, which would protect him in the short term from whatever illness might be around, but it wouldn't confer long-term immunity. You also have to bear in mind that the antibodies can suppress the production of your own antibodies, which is put to good use in treatment for the rhesus negative condition, where antibodies are used to suppress the production of new antibodies. But basically, yes, the answer is it is short term. However, it is potentially an area with a very important future. Before we had antibiotics, plasma treatment was looking like the only way we were going to combat infective disease. And farms were set up to produce large amounts of gamma globulins. And we might be heading back in that direction as we tackle the antibiotic resistance problem.
Okay. Louise has asked, will a single vaccine protect if there are multiple different strains of coronavirus?
Short answer, no. We've already had this experience with influenza, that every year there's a world conference called to work out which strains of influenza to put into the vaccine. So when you go to the surgery or the boots or wherever you go to have your flu jab, what you're being given is, I think it's usually 3 strains, and they've worked out which are the most likely to arise. The coronavirus that we're looking at here, I mean, there are lots of coronavirus. Most coronaviruses that we know about cause common colds. The one that we're looking at now, so far, it doesn't appear that there are other virulent strains arising. There will be mutations. Most mutations are deleterious. In other words, the organism becomes less effective. But the simple answer is you need to have a vaccine to each strain for it to be effective, because only that way will you generate the specific antibodies that will tackle that particular antigen.
Okay, another question from Henry. As regards COVID-19, do you think it's possible that it will burn itself out without the need for a vaccine?
If it kills enough people, yes. I'm being slightly glib there, but on a very serious issue. The thing that's been happening, unfortunately, the world has undertaken a huge experiment. What happens if you get one person infected, who then goes and infects various other people, and so it goes on. What happens is that the people who die are the most vulnerable. And in this case, the most vulnerable are the elderly and people with a whole load of so-called comorbidity. In other words, they've got illnesses which will make them more vulnerable. So if you just let it run its course, then what will happen is that some people will recover. Most people who get this virus have a mild reaction or are even totally unaware, but a significant proportion, I mean, there are 41,000 people in this country who haven't recovered, which is a large number, put it that way without being too dramatic. So what happens, like any disease that runs through a population, the Black Death, it will kill the most vulnerable people. With a virus like this, it is killing mainly the elderly people. So in evolutionary terms, young reproductive people are not so affected, and therefore it won't affect world population in that sense. But when we say a virus runs its course, it's still there. And you only get rid of a virus if you completely remove its ability to grow. The virus is not truly a living organism in the way we think. It can't live on its own. It can only multiply in the body of a host. So if you get to a point where the virus has got no hosts to multiply in, either because they're not there, or because the people, in this case, have immunity, and therefore will kill anything that comes in. So yes, it will run its course, but at huge cost. And it could still get worse, because you think less than 1% of the population has been infected so far. And that means there are plenty of other people around who are now vulnerable.
Tony and Jenny have asked, what is the correct term for an individual virus, body, cell, or entity? Because it's the actual particle.
Yeah, you just call it a virus.
Yeah.
But it's a very good question, because yeah, just we can forget Latin now. The Latins are all gone. So if you just talk about, often you'll say a virus particle to make sure that no one gets confused thinking there's such thing as a virus cell. Virus particle, because it's really just a few substances, a bit of RNA, a bit of protein, a bit of fat.
Moving briefly away from COVID-19, Anne asks, how does hay fever happen, given that the invader is pollen that's been around all my life?
I know, tell me about it, because I get it every year. People told me I'd grow out of it. Well, I'm coming up to my 70th birthday, so I haven't yet grown out of it, so I'm still waiting. What's happening there is that the pollen, when it gets to that first line of defense, so it's not to do with your specific response so much as it will stimulate the cells to produce histamine. And histamine is one of those sort of signaling chemicals which is there to do useful things like making the blood vessels get bigger so that more blood can flow and therefore get white blood cells to the site of the problem more effectively. But unfortunately, it causes us all those symptoms which we find so distressing. It's really bypassing the system. If you're allergic, in my case it's probably Timothy grass, specifically to that, then your system has failed. So I'm confessing to be a bit of a failure. And when I was talking about genetic variability, some of us are susceptible, some of us aren't. And it means that the system isn't working as effectively as you might. But it may be that that evolutionarily, hay fever was linked to something else which is advantageous, like particularly good looks or something like that. So I can console myself in that.
Okay. Tony and Jenny again, if a vaccine is not that effective, can you have several different not so good types? I guess we do that with flu anyway, where we have the trivalent or tetravalent vaccines that are seasonal flu.
Yes, absolutely. It's a constant game that the flu virus keeps on mutating. We have to keep guessing which one is going to become. I made a mistake a few years ago. I went to a conference on the flu vaccine, and we had the world's expert on flu vaccines and how they was explaining how they set up the decision as to what to do. And the room was full of people who are current researchers on flu vaccine. So a week later, I got the flu. But it's a cat and mouse game, this, and you just have to keep trying to do your best to find. Again, the thing to remember with flu is that it is a major killer. We've got used to the idea that COVID is a major killer. Flu itself is a really dangerous disease, and of course, it's what wiped out millions back in 1918.
Robin's asked, does COVID-19 kill us, or is it our reaction to it?
It's always a moot point. Well, it's our reaction to it, effectively, because the virus itself does nothing but mess up your system. So it's all those things. At the extreme, it's when I was describing the cytokine storm, where the system is reacting much too well, and is therefore, you produce lots of mucus, for example, and it drowns you. And that's why it's such a horrible infection. So yes, I mean, when people get an extremely high fever in any illness, that fever is raising the body temperature to a level where the body isn't going to function properly. So in a sense, any illness, the cause of death is your reaction to it. A very good point.
Although I have seen some reports that are suggesting that where the virus targets these two receptors in certain types of cell, particularly in the lungs and cardiac muscle, that they're seeing signs of significant tissue damage there. But is that still the immune response that those cells that have been attacked by the virus are then taken out by the immune system? Exactly.
This is why they have this lovely term, killer cells. There are two types of T cells which are killer cells. One's the T cell which is specific for a particular antigen, and also natural killer cells, or NK cells, which will roam about and knock off anything which is looking dodgy. Another talk that I've given a little while ago, I look at that in the context of cancer, recognizing self and non-self. So the body has to attack itself in order to get rid of the thing that's causing the problem.
And Jones has asked, can you measure?
How are we doing?
Yeah, we've got a few more coming in. If you're still up for it, 2 or 3 more questions.
Go on, carry on. Carry on.
And saying, can you measure the strength of the person's immune system?
In a sense, but you do it by challenging them. And that's what they refer to. You're given a challenge to see if you respond to it. And it appears that some people have more active immune systems than others, but it's so complex. You've got so many factors involved that a general notion that some people are better at being immune than others is difficult. I wouldn't see it myself as generally true. What is true is that people who are young and healthy tend to have immune systems which are more effective than people who are older and unhealthy. One of the things you can do is to make sure you have a healthy diet. I'm not a believer in magic foods, but if you're generally healthy, you're going to be generally healthy.
There seems to be quite a lot of stuff going around at the moment about the value of vitamin D in particular. I guess it's still rather early days to be conclusive about that.
I've got to confess to being a bit of a skeptic about these things. If you have a good diet and you've got the level that you should be having, I mean, vitamin D was another one that people mentioned, but people should be managing their vitamin D in any case for so many other reasons. And if you are unhealthy for any reason, if you've got a vitamin deficiency, which of course is very rare with a normal diet, then you're going to be more susceptible to a whole range of illnesses.
Yeah. Steve's asked, are there scientific bodies continually monitoring new viruses as they appear in the world? Is there a way to create a generic vaccine that just requires a simple modification, fast track vaccine? I think it's two distinct questions there.
Yeah, yeah. The first question is there a body? Yes, the World Health Organization spends a lot of its time doing that, and it issues clear advice and guidance, which some countries follow, and some countries decide to do something else. As far as, is there a sort of, you're really looking for a magic bullet here, a universal vaccine? The answer is no, not going to happen, because you've got to have specific vaccines for specific organisms. And the problem, as we found with COVID and with SARS and MERS, is that if you have a means of generating lots of new viruses, you'll continuously have pandemics.
Yeah. Okay. Linda's asked, if someone has recovered from chronic fatigue syndrome, being left with allergic and autoimmune reactions to several medications, are they being overcautious to shield despite not being on the government list?
I think the watchword is, if you have a range of disorders of that kind, then you need to be cautious, because the evidence is that the risk is highest in people who've got a range of various problems. You know, obesity is a big one, but I think if you've got if your immune system is compromised in any way, then the risk is potentially higher.
Bob, was your question public in the end?
Yes.
Okay. So Bob was asking, why is excess weight a health risk with regards to COVID-19?
Good question, and someone's going to make some money out of explaining it all eventually. There are many factors. One thing is that obesity carries a whole range of other medical risks, and it's well known the link between obesity and type 2 diabetes, which again has various other knock-ons, but also physiological problems to do with how well you can breathe if you are carrying excess weight. So I think really, again, it's one of those things, there are lots of factors involved. And what's happened is we've just looked at the crude evidence, and that obese people are at greater risk. I don't yet know whether that means a greater risk of contracting the illness or at greater risk of it becoming serious because of those associated factors.
So that brings us to the end of the formal meeting. Many thanks to Andrew for an enlightening talk, and to you all for attending. A reminder that if you wish to support what we're doing, you can donate to paypal.me/WinCafe Sci. Details are in the chat somewhere, and this recording will be available on our YouTube channel in the next day or so. So thank you all very much.
And thank everybody.
Great.
Okay.
Thank you, Andrew. Yeah.
Goodbye, everybody.